Imagine walking into any biology lab in the world today. You'll see scientists ordering custom-made genes the way you might order a book from Amazon. They type a DNA sequence into a website, click Order, and a few days later, a package arrives with exactly the genetic material they requested. This multi-billion-dollar industry, the foundation of gene therapy, CRISPR, and modern biotechnology, exists because one man, born in a village with no school, no electricity, and not even a pencil to his name, decided to ask, can we write the language of life ourselves? Har Gobind Khorana, the poet of the genetic code. January 9th, 1922. Raipur, a tiny village in Punjab, British India, now part of Pakistan. A son is born to Ganpatrai Khorana, a village agricultural taxation clerk, and his wife, Krishna Devi. They name him Har Gobind. He's the youngest of 5 children. Here's what you need to understand about this place. Raipur had about 100 people. The Khorana family was practically the only literate family in the entire village. Although poor, my father was dedicated to educating his children, Har Gobind Khorana said. For the first 4 years of his education, young Har Gobind sat under a tree. That tree was the school. There were no desks, no blackboards, no textbooks. He didn't own a pencil until he was 6 years old. Think about that for a moment. A boy who couldn't afford a pencil would one day write the very alphabet of life itself. But young Khorana had something more valuable than a pencil. He had an unquenchable thirst for knowledge. He walked to school, he studied under street lamps, he read every book he could borrow. By the 1940s, he'd made his way to Punjab University in Lahore, surviving on scholarships. In 1943, he earned his bachelor's degree. In 1945, his master's. Then came 1945. World War II was ending. India was on the brink of independence and partition. Khorana received a Government of India fellowship to study in England. He boarded a ship and sailed for Liverpool. I didn't have a clear career plan. When I applied to university, I actually applied to 2 departments, English literature and chemistry. I had no idea where life would take me. At Liverpool, he studied organic chemistry, earning his PhD in 1948. Then came a postdoctoral year in Switzerland, working with the legendary Vladimir Prelog at ETH Zurich. This Swiss year would prove fateful, not just for science, but for his heart. He met a woman named Esther Elizabeth Sibler. They would marry in 1952 and remain together for nearly 40 decades. In 1950, Khorana moved to Cambridge University on a fellowship, and this was the moment that changed everything. Cambridge in the early 1950s was the epicenter of a scientific revolution. Frederick Sanger was figuring out how to sequence proteins. Max Perutz and John Kendrew were unlocking the structure of hemoglobin. And in a nearby lab, 2 young scientists named James Watson and Francis Crick were quietly working on something that would shock the world: the structure of DNA. They published their discovery in 1953. Khorana later wrote, "Cambridge was the only inspiring place. Molecular biology was being born." He worked under Sir Alexander Todd, who was unraveling the chemistry of nucleic acids. The seeds were planted. Khorana became fascinated with these molecules, DNA, RNA, that carried the secrets of life. But first, he needed a place to build his own lab, and that place turned out to be Canada. In 1952, Khorana moved his young family, now including his wife Esther, to Vancouver, British Columbia. He joined the British Columbia Research Council at the University of British Columbia. The facilities were modest. Actually, that's generous. They were minimal, but the council gave him priceless freedom. They gave him all the freedom in the world. Khorana began experimenting with chemicals called carbodiamides, compounds he'd encountered by chance in Switzerland. He used them to synthesize molecules that biochemists desperately needed: nucleotides, coenzymes, the building blocks of nucleic acids. Word spread. In 1956, 2 giants of the emerging field, Arthur Kornberg and Paul Berg, visited his tiny Vancouver lab.
Kornberg would later win a Nobel Prize for discovering how DNA is replicated. Berg would win one for developing recombinant DNA technology, and both came to see Khorana. Khorana realized he needed to learn biochemistry properly, so he did something humble and brilliant. He went to Kornberg's lab to study. He later said he wanted to learn from the master. By the 1960s, his reputation was established, and a powerhouse American university came calling. In 1960, Har Gobind Khorana moved to the University of Wisconsin-Madison as co-director of the Institute for Enzyme Research. Within 2 years, he became a full professor of biochemistry. He was about to enter the most exciting scientific race of the 20th century. Here's what scientists knew. DNA contains 4 chemical letters, A, T, G, C. These letters somehow spell out instructions for building proteins, which are made of 20 different amino acids. But how? How do 4 letters code for 20 amino acids? It's like having a 4-letter alphabet that needed to write a 20-word vocabulary. The answer, proposed by physicists and cryptographers, was triplets. 3 DNA letters, a codon, codes for 1 amino acid. 4 letters, taken 3 at a time, gives you 64 possible combinations, more than enough. But which triplets code for which amino acids? That was the puzzle. And in 1961, Marshall Nirenberg dropped a bombshell. He showed that a string of UUU, all 1 letter, produced a protein made entirely of 1 amino acid, phenylalanine. The race was on to crack the entire genetic code. Khorana entered the race with a unique strategy. Instead of using random RNA sequences, he decided to synthesize RNA with precise repeating patterns. This was painstaking work. Remember, this was before computers, before automated DNA synthesizers, before everything we take for granted. Khorana built these molecules by hand, using organic chemistry. He made RNA with 2 repeating units, UC, 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 UC. When he used this to direct protein synthesis, he got alternating amino acids, serine, leucine, serine, leucine. That told him UC2 codes for serine, CUC codes for leucine. He made RNA with 3 repeating units, UAC, 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 and got 3 different amino acid strings, proving that the code is read in triplets without overlap. He made RNA with 4 repeating units, UAG, UAA, UGA, and got nothing, just short fragments. He had discovered the stop signals, the punctuation marks that tell the cell the protein is finished. Piece by piece, codon by codon, Khorana and his team assembled the entire genetic dictionary. They proved that the language of life is spelled out in 3-letter words. He made important contributions to this field by building different RNA chains with the help of enzymes. Using these enzymes, he was able to produce proteins. The amino acid sequences of these proteins then solved the rest of the puzzle. On December 12, 1968, Har Gobind Khorana stood in Stockholm, Sweden, to receive the Nobel Prize in Physiology and Medicine. He shared it with Marshall Nirenberg and Robert Hawley. The boy who couldn't afford a pencil had written his name in the history books. For their interpretation of the genetic code and its function in protein synthesis, Nobel Prize citation, 1968. But Khorana wasn't finished. In fact, he considered the Nobel Prize just a milestone. In 1970, Khorana moved to the Massachusetts Institute of Technology, MIT, as the Alfred Sloan Professor of Biology and Chemistry. And he set himself an audacious goal: synthesize an entire gene from scratch. Remember, at this time, scientists could barely sequence short pieces of DNA. Khorana wanted to build a functioning gene, not copy one from nature, build it chemically, letter by letter. In 1970, he and his team announced success, the first complete synthesis of a yeast gene. By 1976, their team had gone further. They built a gene that could actually function inside a living cell. Think about what this meant. For the first time in history, humans were not just reading the book of life, we were writing new pages. The methods Khorana developed became the foundation of an entire industry. Today, anyone can order synthetic genes online. Scientists use them to create insulin, to engineer crops, to develop gene therapies, to understand diseases. Khorana's work also anticipated the invention of PCR, polymerase chain reaction, the technique that copies DNA millions of times, essential for COVID testing, forensics, and basically all modern molecular biology. After conquering the genetic code, after building the first synthetic gene, Khorana did something unexpected. He switched fields entirely. In the mid-1970s, he became fascinated by how we see. Later, he explained, many of us felt that after deciphering the genetic code, an era ended. The new frontier could only be the